Okay, I will. I have 95 slides here. I'm not sure I'm going to get through all of them, but we'll see. You might have a bias. I'm always a bit hesitant about those bios because they make us sound so senior. And I, sadly, by the color of my hair, I am getting more senior. But not that many years ago. Well, that many years ago, but 25 years ago or so, I was roughly in your career age now. So I did a PhD in theoretical physics, and then more or less by necessity started hacking codes because I was doing simulations of membranes, which was really, really difficult at the time. I'll come back to that. And then somehow, when nobody else could fix the code, I ended up fixing it. And after a few years, I'm not quite sure it happened, suddenly I was leading the entire code team. And yeah, that's kind of the story of my life. So nowadays, GROMACS is developed in my team. But as I mentioned, I've benefited tremendously from this, from standing on two legs, where one leg is my scientific application, explaining membrane proteins with any methods available. And nowadays, half of my lab is experimental, actually. We do a lot of cryo-electron microscopy. While on the other hand, we have the HPC methods coding, and then I also have a gigantic group, both working on codes, simulation approaches. And just getting these two teams to talk to each other is great fun. And for me personally, I find it awesome that I can both go to Biophysical Society in February here and talk to my colleagues in biophysics, but then I can be at a meeting like this, talk to people solving other problems, maybe CFD come in, and focus on the computational part and trying to change the state of the art of computing in the world, not just in life science. And then more or less by mistake, because I kept having opinions about everything, suddenly they asked me to take over the directorship of the Swedish National High Performance Computing Infrastructure last year, which is fun in its own way, but it takes a lot of time. But I'm going to bring this up from another point of view. 1991, I had just finished upper secondary school, and I have no idea what I wanted to do with my life. There are two things. I loved computers, and I somehow loved life science. And this was the hardest decision in my life, deciding what to do. Now, you're fairly young. So in 1991, yes, that's, in hindsight, that was the last year of the Soviet Union. So sadly, I am that old. On Swedish television, Eurovision, that was the age of the broad shoulders and everything. I think this fashion is coming back now. And you also got these amazing new lightweight portable cell phones. A few things have changed since then. At the time, I applied and got into the very prestigious MD PhD program at Sweden's most famous medical hospital. They hand out the Nobel Prize for Medicine. And I realized after 1 week, I can't stand this. Because I'm a bit lazy. And apparently doing an MD, that is about learning 10,000 pages by heart every year. While studying physics, on the other hand, then you spend 6 weeks learning a book with 25 pages. You just have to think. And that is a much better fit for somebody who's a bit lazy but likes to think. So I abandoned the MD track, which tormented me. I had no idea whether that was right, but really went more into pure life science and understanding physics, everything. The rest of my family, they're MDs. And this is a story you can think back a few hundred years. So how do we see and understand life? None of you have seen any of these proteins or atoms or anything that you work so much with. They're kind of not quite make-believe, but almost. So the very first microscopes that were developed by Anton van Leeuwenhoek in the 1600s. And the whole idea is that you put a small sample there, and then by holding this up to a light, you could start to see the detailed, well, somewhat detailed, like resolution 0.1 millimeter or so. And this led to a bunch of discoveries such as cells and tissues. Of course, you didn't have cameras on the time, so they had to draw all these things. In Sweden, in the 1800s, Jöns Jacob Berzelius, who actually funded that Karolinska Institute, he tested different types of food in animals and realized that if you give dogs, I think it was, if they only get carbohydrates, they die. And if they only get fat, they die. But if you feed them what we nowadays call proteins, so eggs, they can actually survive. And that's what the process for the primary source of food. And of course, you are educated, so nowadays you know what these proteins consist of and everything, and we're skipping a few hundred years ahead there, but proteins have always been the passion of my life. And there are many ways you can address proteins. Ultimately, it comes down to chemical molecules. And when we're talking about chemical molecules, one obvious way of doing this is with quantum mechanics. I'm not going to talk about quantum mechanics here. It's an excellent method in many ways, but as I'm going to show you in a few slides, it also has some fundamental problems. Not if you're looking at molecules like this, but if you're looking at proteins. The other important development that happened in the 1900s was that Fred Sanger started to sequence proteins first. This was a gigantic tour de force. So he probably spent like 10 or 15 years sequencing, getting the sequence of insulin. And I won't have time to share you exactly how I went through this. But he was a very lonely wolf, did this almost entirely alone, and eventually came up with, published this sequence with 58 amino acids. And that, of course, shows that a protein, the sequence in a protein is unique, and nowadays you know the connection to DNA and everything. And to me, this is a fascinating interplay between life, chemistry, and physics. I'm not sure if you've seen Sidney Harris. He used to make all these cartoons for Scientific American that I'm on the verge of a major breakthrough, but I'm also at that point where chemistry leaves and physics begins, so I'll have to drop the whole thing, which is kind of the curse of not just my life, but many scientists. And I'm not going to take you through the entire history of life science, but over the decades here, Rosalind Franklin determined the first X-ray scattering patterns of DNA that led to Watson and Crick deciphering the molecular structure of DNA, etc. And nowadays, in the since the 60s, we can build entire molecules of these amazing proteins. If you ever go to Cambridge, this model is still present in the hallway of the laboratory there. So everything is built manually. And it took them 22 years to determine these first structures of proteins. The amount of work that went into this, calculating each and every coordinate in detail. But in the 1970s, when we started getting more and more of these sequences, Helen Berman in particular, they started to collect coordinates of these proteins in databases. Oops, sorry. No, I think it was fine. It was just a blank slide. Turn it on again. There you go. And that's, if you've ever worked with proteins, you've seen these entries in the protein data bank. And this is important because now all these things in life science, they are now turning into numbers. Kind of similar way bioinformatics is turning biological evolution and everything into statistics about the sequence variability or so. And of course, 1960s was early, but once you can turn this into numbers, you can start applying physical models to it. And here too, I'm going to need to skip a bunch of things, but there were a number of groups around Schneider-Lipson and everything. They basically, partly because they were chemists and not physicists, instead of using quantum mechanics, they ended up with very, very simple interaction models and simple atoms, and then figured, is there any way, in principle, if we have all these atoms, we should be able to model the entire interactions of them. This is lysozyme protein. And I won't have time to go into all the details of how you model this, but even with X-ray in the 1960s, they started to have the first computers. So they actually tried to minimize these models that people had built manually to see, can we improve them? And this is pretty much the birth of using computers in life science. And we're going to need to jump a little bit, but before I do that, I will actually show one of the first movies here of my postdoc mentor, Mike Levitt, where they did some of the first ever molecular dynamic simulations of the bovine pancreatic trypsin inhibitor, a tiny protein by today's standards. And I think this movie was, all the titles here are longer than the actual movie. But this was a revelation to experiments, because prior to this, in experimental science, we had X-rays crystals, right? That from an experimental point of view, we always saw proteins, they always saw proteins as rigid. And as you're going to see here in a second here, what these simulations actually made people realize, at room temperature in particular, these are not rigid molecules. They're moving. Now, this could, of course, be an error. I would argue that the real fame of these methods came when we realized that if these molecules are moving, we would expect there to be a little bit of motion blur in all the X-rays, so broadening of atoms. And this is what today is called B factors. You can actually see this broadening in the X-ray structures, which is the reason we even believe this in the first place. And now I'm going to jump to another bunch of things, and that's kind of me starting. So once you know this, you could in principle start to run a simulation of anything. This is what I did when I was a PhD student. I ran gigantic simulations of a large membrane. So the reason I'm showing you this is that this is so what you should not do in simulations. Because I came in, I was enthusiastic, my advisor was away for a sabbatical 9 months. So you take a gigantic system, you put it in a computer, and you push the big red button, and you wait for something interesting to happen. If you've done that, well, you're in good company. I say, I've done it a few too many times too. The problem is that there is a very big risk that nothing interesting happens. And then you might have wasted 6 months. In this particular case, I was lucky. Actually, we did see some fun things, and it had to do with the buckling of these membranes. And this buckling, you can actually both relate to physics, and for lipids in particular, that means that you can connect motions all the way from the atoms all the way up to cells. So I was a bit lucky, but I figured that you can't always be lucky, so I'm going to try to share some of these experiences with you. The first thing you need to do to avoid being lucky, there is this famous quote by Richard Feynman that your first task as a scientist is to not fool yourself, and you're the easiest person to fool. You've probably seen this Magritte, that's the cine pipe, that this is not a pipe. Actually, and the reason for that, it's a painting of a pipe, and this is not even a painting of a pipe. This is a JPEG image of a painting of a pipe that you're seeing as electrons, etc., etc., etc. The point is that this is not a protein. This is an illustration of a computer trying to interpret physical equations that we have used to model something that we might think is reasonably close to a protein. So your first task in life science is that don't get too carried away with the computers. You need to focus on the life science problem, and you need to focus what is it actually you're trying to solve in life science. So don't do what the junior version of me did, that you had a very large computer, so you're starting something and waiting for something interesting to happen. That is not the life science question. So how do we approach life science questions? Well, couldn't you just try to determine the quantum chemical, just solve the wave function of that protein on the right? The problem here is that it's moving, right? So that for any of these frames, in theory, you could solve the wave function. But the wave function has some other problems that normally use a Born-Oppenheimer approximation. If you don't know what it is, don't worry. But that basically means that you're assuming that atoms don't move. What temperature is it if atoms don't move? 0 Kelvin. So how many diseases happen at 0 Kelvin? At 0 Kelvin, we are all dead. So studying life science at 0 Kelvin is a pretty bad approximation to start with. The other problem, if you're going to do quantum chemistry, you can't afford to have all the water. So people ignore the water. So with quantum chemistry is an amazing method if you want to study things in vacuum at 0 Kelvin. But there is not a whole lot of life going on in outer space. So I would suggest we ignore that, and that's why I mostly ignore quantum chemistry. The person behind this, and now we're gradually getting to that, was Christian Amundsen, who had this. Do you know what Christian Amundsen came up with? That's great. I would argue the greatest discoveries are frequently the ones that 50, 60 years later are obvious. So Christian Amundsen came up with this hypothesis that the native state of a protein is the thermodynamic minimum of free energy. So that's the whole point. That's decided by physics. A molecule will spontaneously fold into the shape that has the lowest free energy. Now, how many of you are physicists? Okay, almost none. So in physics, free energy is a combination of two parts. E, which is just the energy that you could calculate with quantum chemistry. But then you have entropy on the, let's see, on your right here. And you can't really calculate the entropy. The only way to calculate the entropy is to sample all states. So most MD simulations, we actually do not do it just to reach a microsecond or a millisecond or something. Molecular dynamics simulations are amazingly good at sampling the entropy on the right. And while they are certainly nowhere near as good as quantum chemistry when it comes to calculating the exact potential, well, quantum chemistry calculates one frame where atoms are not moving. Molecular simulations, we can easily calculate 1 billion frames at room temperature when things are moving. So they're not perfect, but they're much better at handling this sampling realistic large-scale systems and accounting for free energy. And another way of seeing free energy is that if you're comparing, this is actually not my kids' room, my kids are older nowadays, but a kid's room on the left, that is one state, and that is one state. Now, those of you who have kids will realize that the probability that it's going to be on the right is virtually zero. But in principle, each of these states is just one state, but there are many, many more states roughly like the one on the left, while only one on the right. And entropy really deals with all this stuff, handling numerous states. If you want to go more into details about this, there is an entire 13-lecture class I've recorded on YouTube on molecular biophysics that you can look into. And this finally brings us to molecular dynamics and molecular simulations. So if we now have one of these systems, the way we try to calculate this is that in this case, we want to sample millions or billions of conformations on the system and also evolve it over time. And that means that I can't afford to have a very fancy interaction that would take 10 seconds to calculate because I would only be able to afford a few thousand frames. This needs to calculate in a millisecond. So what you do is that, well, we pretty much pick very simple potentials for bonds and angles, and these might not look so simple, but the whole point, they're just harmonic. They're like small springs. The amazing part here is that all these coefficients we get just by fitting things to reproduce experimental results, which is what the Nobel Prize to Levitt and Ari Warshel and Martin Karplus was roughly awarded to 10 years ago. Now, the first four terms here are not that important because there are very few of them. There are not that many bonds in the proteins. But at the very end, you have two terms here, which is the sum over all pairs, where the first one with the Q's, that's electrostatics, and the lowest one has to do with so-called weak non-bonded Lennard-Jones interactions. And that's a pain, because if each atom here is surrounded by maybe, well, there might be 500 atoms within 10 angstrom. You need to, for every time step, you need to calculate these interactions with 500 other atoms for each atom. And that's a lot of floating point operations. That was completely not possible in the 1960s. But if you could do that, again, we're skipping the fancy quantum mechanics here, then we have the force on each atom. And if I have those forces, well, then I can do the acceleration. That's just the force divided by the mass on each atom. And then you take, if we know the acceleration, I can calculate how the velocity is changing. And if I know how the velocity is, I can calculate how the position is changing. This is easy as pie. The only problem is that you need to take very, very short time steps here, 1 or 2 femtoseconds. So you're going to need like 10 to the power of 12 steps, 10 to the power of 15 steps to get to a second, which we won't. But over some sort of scale, this generates a trajectory, and I can show you one for a small motor. I know, you're in shock and awe about how much we're learning about life science here, right? So this worked for physics. As you see, 50 femtoseconds is nothing. But the cool thing that even on my laptop, I could probably do 100 nanoseconds in less than the time it takes me to deliver this talk. And on a supercomputer, you can do 100 nanoseconds for a large protein in an hour, or 1 or 2 microseconds per day. So the reason for moving to these larger and larger computers is that when I did my PhD, these were methods that roughly the cat would have dragged in. Nobody was interested in it. But what happened some 20 years ago, roughly, is that biochemists realized, wait a second, are you saying that you can calculate, say, how an ion is passing through an ion channel? You can calculate how small molecules are binding? So this effectively became a computational microscope to biochemists. And this suddenly made the entire field take off. So I, who have a background as a physicist, today there are like, I think there are 10,000 papers per year published using GROMACS. Because everyone in life sciences uses this method. And there are many other programs than GROMACS too. So with that, does that work? Does that mean that we just take a protein in a computer, push a button and they simulate it, and that means that we can predict what's going to happen to that protein? Well, the problem is that physics, this comes down to what a simulation is. If you're studying mathematics and chaotic systems, this is a chaotic system, and the formal way of defining that is that it has so-called positive Lyapunov exponent. What that means is that if you have a very small error in the initial conditions, the trajectories will diverge from each other. So that's bad. Because it means that you can never ever determine a structure exactly, and we don't know what the initial velocities are. I can just assign those from average velocities I would expect at room temperature. But that means that it's impossible for me to ever predict how a molecule moves, which is a bummer. So why are we doing this in the first place? Well, the first thing is that if you look at this prediction from Hurricane Sandy in weather simulation, it's roughly the same thing there, right? That each individual trajectory has errors. But if the average of this 100 trajectories or so end up pretty much straight on New York City, and that's where the hurricane hit. So the cool thing is that you can apply statistical mechanics concepts that on average we know what these trajectories will be. The other point is that 3 or 4 days in the future, the uncertainty is large. But over the next 24 hours or so, the uncertainty is fairly low here, right? So you can formulate this in ways showing that so-called shadow trajectories, that although each individual trajectory is not an exact protein, but over reasonably short timescales, you know, the time it might take an ion to bind if this is a protein or something, I know that I'm going to be very, very close. Even each individual trajectory is likely to be very close to the true motion. And if I then execute many of them, I will be able to apply uncertainty quantification and statistical methods to actually handle the average. And this is why we can fold proteins, for instance. You can do this experiment yourself. If you just touch yourself, you're not going to die. But of course, you change the boundary conditions, right? You change the trajectory of some proteins. But life in general is pretty stable. So the way we then handle that, if this idea works, we would like to sample as much as possible and learn as much life science as possible. And then many people, me included, you will have seen some amazing fancy simulations of, well, either entire cells, gigantic vesicles. Visualization centers and supercomputing centers, they love these, but because they look fantastic on covers of magazines or as an image in a visualization or something. But that's not the science. So the science, if you want to sample a system really well and you want to reach long time scales, you want to pick a small system. The smaller your system is, the better you can sample it, and the cheaper it's going to be to sample. So the one thing I would encourage you to be boring. Start, before you even start that simulation, consider what is the absolutely smallest system you can imagine that would actually reproduce this. Because if you have a tiny system, they just say a small protein or something, you might be able to reach 100 microseconds per day where you effectively reproduce biochemistry experiments. Now, if you're picking an entire cell, you're not going to reach 1 microsecond per day. You might be at 1 nanosecond per day, and you're not really doing biology. You're just drawing fancy images. So think about all these things before you even consider hitting that red button. What is the system? Don't go for a large system because it's cool. Go to the smallest system possible that you think can model. Remember, here's where you apply your judgment, right? You are the person who decides how simple your model should be. You should always simplify as much as possible, but never more. And then make a realistic guess. Based on what you know about this process, don't just hit the red button because you can simulate 10 microseconds. What if the process you're studying requires a millisecond? Simulating 10 microseconds is pointless. You're a factor 100 too slow. So this is the other boring part. Go to the library. Read up on this. That roughly, if you're talking about ions being conducted in an ion channel, you can estimate what the current is. And then you can estimate how long should it take for each ion to pass through an ion channel. So make a so-called back of the envelope calculation. Is there any chance that you would be able to model this process, say, over 10 microseconds or so? And if you think there is, and you know that you can simulate those, that's when you start thinking about simulating. The other problem is that what we typically start with is some small molecule here without water. That's not going to work. Quantum chemists frequently stop there. The first thing we need is water. Otherwise, nothing in biology will happen. But now we have these boundaries on the left and right? So that in theory, you could have a sphere here or something, but that is complicated for other reasons. So you typically apply some sort of boundary condition, and the plain simple boundary conditions in simulations, when things go out on the left, they appear on the right. So you effectively have an infinite number of repeated copies. And at some point, you can make this very, very complicated. This is a large membrane protein in a membrane with water, with ions and everything around it. And my reason for mentioning this, many proteins are not stable unless you have, for instance, the correct composition of a membrane or the correct concentrations of ions in a system. So if you're trying to fold the protein, and this protein only folds if you have 100 millimolar of sodium chloride, you do not have any ions in your simulations. It's pointless to even try. You're going to try to reproduce something that this would never happen in nature. And I think I was about to say my students, but I should rather say me. I've done this mistake so many times in my career that you just go too quickly into the computer and press the big button and hope that we're not going to brute force this in a computer. But biology is a bit harder than that. We also need to control things like temperature, which we can do by just adjusting the velocities of the atoms, scaling it up or down a bit. And we can control the pressure by sizing the box up and down a bit. And in any program such as GROMACS, there are plenty of settings when you start a simulation here. My only reason for not going through that is that it's not obvious that you have to use GROMACS. There are plenty of other good tools too. And these particular details, how do I adjust the temperature in a simulation, we have excellent tutorials online for. But I figured I'm going to show you a few examples here, just to get you an idea what we're working on and what one can do. So my favorite application problem is ion channels, and in particular the ion channels in the nervous system. So when nerve signals are typically conducted in a nerve cell with by voltages, and when they get to the end of a nerve cell, the influx of calcium here means that these small synaptic vesicles, they merge with the plasma membrane here, and then they release the small, atoms here. There can be amino acids or so-called neurotransmitters, and they diffuse over a distance here that might be 10 micrometers or so. And if the right neurotransmitter finds the right receptor here, these receptors open an ion pore here, and then they let ions through into the next nerve cell. And depending on what neurotransmitter this is and what channel it is, it's either going to be positive or negative ions. So they can either create a new nerve impulse or inhibit a new nerve impulse. And these are models that we've long tried to study, both experimentally and with computers. So the question is, can you simulate this? Well, hard. The entire cycle here would take in the ballpark of half a millisecond or something, so no, we're not even going to try that. But what we can try is looking at the details of this particular system. For instance, what happens if you're binding small drugs to this, and where do the ions go? These are modulated by a number of drugs, such as anesthetics, alcohol, benzodiazepines, and everything. So what already 10 years ago, I had postdocs doing simulations of this in an entire membrane and everything. But rather than just doing one big simulation hoping to see something, we ran hundreds of simulations. And then you do statistics and check, in this case it's ethanol, but where on average does ethanol end up? And all these different colors here, in particular the blue and yellow, are binding sites where we see significant density of ethanol. So the key message, I calculate average, right? I do a statistical analysis. Rather than looking at how one ethanol molecule is moving, I calculate on average, where do the ethanol spend time? So if you look at one molecule alone, it would look like on the right here, right? It's moving around lots of different conformations. Although I'm pretty happy with this motion because this molecule is located right next to a residue which happens to be a serine 267. And we know that if we change that residue, the molecules no longer bind ethanol. So to me, as a scientist, this is a way of checking the simulations. I see this in a computer, but this makes sense. It matches something that other people have seen in the literature. If this is the binding site, and this is exactly right next to a residue that abolishes binding if I change it. Now, that is not a guarantee this is correct, but now the simulation supports the experiment, and the experiment supports the simulation. And over the years, I would actually argue that most simulations I do today are simpler than the ones I did 15 years ago, because we're always trying to match them with experiments to have solid results from two different techniques, rather than betting everything on one big hero simulation. But the other cool thing that what you see here on the left, right, is that this is not just a matter of one molecule. On average, we have high density in a number of places, and in this case, there are 5 copies. There are 5 subunits here, and we see good density in all of them. And that also means it's no longer a matter of those fluctuations. I don't have to worry about the fact that each trajectory here is not exact, because it's very clear I see a recurring pattern here. And again, if once I see all these blue ones, I don't really worry anymore about what is the length of my simulations, do I have exhaustive sampling. This is a very strong signal, and it also turned out to be correct. Now, once you do that and have that testing, then you can of course start looking at how individual molecules are moving and how the red dots here is basically a function of time showing how the molecules are moving into a binding site. Now, to me, this is cool, but you have no idea how cool an experimentalist thinks this is. Because to them, this is the thing they can never see. They can determine, at the time it was X-ray structures, nowadays everyone is doing cryo-EM. But suddenly you have a method that can actually show, like putting flags on atoms and showing how they diffuse into a specific binding site. Not just where they end up, but also the path they take to get there. And Alexandre Bonvin is later today, I think, going to give a talk about how you can use so-called docking methods to very quickly predict where these molecules are going to bind, much faster than MD simulations. So how did we use these simulations? Well, in this case, again, since we're gradually getting better at not just doing hero simulations, we realized that based on where we saw things binding here, that was very close to a residue called phenylalanine 238. And then we start looking at not just one protein here, but comparing different proteins in humans. GLIC is a bacterial protein, the acetylcholine receptor, the glycine receptor, GABA is very important for anesthetics. And around these helices lining the spore here, there are a number of positions here where we see patterns, and I won't have time to go into the details about the specific patterns here. But now I have identified a binding site, and I have a rough idea how my molecules are binding there. Then we go to the experimental colleagues and say, look, based on what we have seen, if you change that phenylalanine in the bacterial protein, that's the phenylalanine, but it's not the phenylalanine in several of the eukaryotic ones here. So why don't you try to remove the phenylalanine? And I'm not sure how much you remember of amino acids, but phenylalanine has a fairly big ring, right? So our guess was that this ring is taking up a lot of space. So if we change this ring to say isoleucine or leucine or even alanine, we would likely make a better binding site, and this molecule could bind ethanol easier. That was a pure guess. Well, not a pure guess. It's a bit of a guess. It's an hypothesis based on what we've seen in the simulation. But the neat thing is that once you have such an hypothesis, this is something you can test in the lab. Now, of course, they can't see it in the lab. The only thing they can see in the lab is, will this conduct more ions? Will this make this protein more sensitive to ethanol in this case? And to make a long story short, yes, that is the case. So then we're starting from experimental data. We're bringing in some simulations and using that to look at it, understand. Based on that, I make predictions that, well, I think I have a model for how this works, but this is still a guess. But if my guess is correct, we should be able to go into the lab, change some things, and then I make a prediction that if you make that change, you should either conduct more ions or conduct less ions. And the cool thing is that if you can then show that, again, you haven't proven yet your model is correct, but this is an instant, fairly high impact scientific publication. The other nice thing is that you tend to get very clear answers. It's either A or B. Either you confirm your hypothesis or you reject your hypothesis. Compare that to those simulations I did when I was a PhD student. Just push the big red button and wait 9 months and hope you're lucky. If you're not lucky, you're not going to get a paper. In this case, we confirmed it. We would have been able to publish this even if it turned out not to be the case. So this is how one should do it. What even I am doing, so once you have very large computers such as Fugaku or so, you can imagine you could simulate a gigantic system. This is very tempting, and it's so tempting that I'm even doing this. I'm going to come back to this. This is a whole cell, very minimal cell. But remember these things I said about free energy. This cell has 800 million atoms. To sample 800 million atoms, you're going to need significantly more than 1 billion time steps. Because again, that if you talk about the degrees of freedom is roughly 3 times the number. You have X, Y, Z, and then actually you would have velocities too. But if you have 1 billion atoms, you probably need 10 billion time steps to even be close to sample it. And 10 billion time steps is a lot. So there are so many simulations that they don't even have a chance to sample things. So that I have done this a few times, but the first thing I want you to do not try to brute force simulations. You need to have an idea what are the timescales involved. Is it realistic for you to sample this? Don't just try to push it. I've seen so many students and postdocs fail just by trying to push big simulations. And again, that's not limited to what we do. So how do you get around this instead of pushing? This is not limited to life science, but all these large problems that you want to sample something or so. Well, you need to be smart. So remember those proteins I showed you? If I completely simulated it, that would essentially be both folding and unfolding the protein, sampling every possible conformation a molecule could ever be in. Sure, that would be fantastic if I could. But that's not really my goal. At least in most cases, that's not the goal. If I'm interested in saying, how does this ion channel open? That's a much more restricted question. In principle, that's like opening a door. If I want to sampling the opening of the door here, I don't need to sample the entire building, right? We just need to sample this door. And then we could, in principle, focus the sampling on this door. And the equivalent thing in a simulation would be using some sort of metadynamics. And I'm going to go through this fairly quickly just to give you the idea, because this is actually a very, very simple idea that people make sound very complicated. In some cases, we know exactly what the process is, say the door. And with the door here, I could, sample here and then sample here, and I could exactly force the day I want it to be and make sure that we've sampled every single conformation of the door, and then somehow compensate for these forces I added. In general, of course, I don't know what these potentials or something are. So in general, if I already know exactly what I'm going to sample and how, then I probably don't even need the simulation. So the problem is that I have some sort of rough process, maybe that it should be an opening of the door, but I don't really know where to start. So the idea with metadynamics is that if we have the blue curve here is a Hamiltonian, but this just really means a potential energy function. So with this potential energy function, that means that it's low free energy here, so the molecule is going to want to spend a lot of time here, and it's going to want to spend a lot of time here. It does not like to spend time here, and does not like to spend time here. Now, the problem is I don't know what that one is, because that's free energy. I can calculate the potential, but I cannot calculate the free energy. But if I just start simulating this, you will very quickly see that, the atom is going to be here, right? And then it just keeps being here. It does not move. That's a bit boring. But if the molecule keeps being here, how about if I try to push it away? Again, I don't know what the blue curve here is, but if I know that it's spending a lot of time here, I know that by definition I must be close to a minimum. And let's just add some energy to push it away from that minimum. And I can show you an image of that. That's probably a movie of that. That's going to be easier. So we start here. The green curve here is the actual curve I'm simulating. This is my small particle, but I don't know what the green curve is. But what I do know, as I'm running the simulation, I can calculate where I am. And these blue bars is basically just the count, how frequently I've been here. And as you see the count, you're going to see that I'm adding a small potential here. And adding the small potential I can do. So as we're doing the simulation, you see what happens when I'm adding the potential here, I will push it away from that minimum. Now, the problem here is, of course, that I've now destroyed my potential. This is no longer the correct potential. I've added an extra potential here, so I'm not really simulating the real thing. But I know what potential I've added here. The potential I've added here, because again, this is just equations. I've added small Gaussians or something. So I can subtract that at the end. And when I compensate for that, I can actually recover this potential, the entire potential, including the very high free energy parts here that I've not sampled. So it's a really nice way to sample a complicated system that would take far too long. So what's the problem with this? Why don't we do this all the time? Well, the problem here, this is a one-dimensional thing. This works if we know that we're interested in the door. then I could say that the only degree of freedom that matters here is how open the door is, right? The only problem, maybe the problem was the other door. And now I've just chosen to restrict my sampling to the complete incorrect part of the system. So here again, your biological knowledge is important. You need to read, if you want to be good at computers and life science, you need to go to the library and read papers. And you need to read papers about failures. You need to understand these things. And you need to particularly understand, if you're studying a particular protein, you should have read 50 papers on that protein, because that is what helps you pick the right part of the molecule to study or so. And unfortunately, no supercomputer in the world can compensate for that knowledge. Another possible, very popular approach that you might see, the so-called coarse graining. Have any of you used coarse graining? Yes? Sorry?
In metadynamic simulation, how do we know the system when it reaches the equilibrium? Like how long we should simulate, for example?
So that's a good question. How do you know when the system has reached equilibrium? And I would say that when the distribution no longer changes. So you remember those? You saw all those blue bars, right? When the shape of this no longer changes, so that we keep adding points equally over the entire distribution, because then I have reached a point where it has converged. So that when let's see here. You start here, you see it's changing, it's changing, it's changing, the shape is changing, and now we're starting to we're starting to sample new parts. But somewhere here, when we're getting to the point where this total curve here is roughly flat, when this curve is roughly flat, I'm going to the red particle will start to spend as much time across the entire part here. And that you can detect. So when you're spending roughly the same amount of time all over this so-called collective variable, that is when you have converged. The other approach that works surprisingly well is that you can cheat. There is not a law of nature that we have that the unit we need to simulate must be atoms. Because again, in quantum mechanics, it would be electrons, right? So instead of atoms, maybe cheat and invent some larger particle. And so this is a gigantic system, and I mentioned we could simulate these membranes for a microsecond or so. But instead of having every single atom in a lipid, turn a lipid into not really atoms, but some beads or particles. So you only have, say, 10 particles in a lipid. Now, these aren't atoms. Same thing for a protein. Every amino acid here might be 2 or 3 particles. This is wrong. You can't really do this. But what I'm effectively doing, I'm encoding some of the stuff would be in all these different degrees of freedom, and effectively moving those over to adding extra terms to this potential to try to describe it. There are errors here, but the cool thing is that the timescales we can then reach are in the ballpark of, like, in this case, just 450 microsecond simulations run on a few desktops. So you can reach orders of magnitude longer timescales with coarse graining. Now, there is no such thing as a free lunch. So the problem with coarse graining is that we might screw up detailed interactions. A normal MD simulation might give you an accuracy of 1 to 2 kilojoule per mole. With coarse graining, I would be very happy if I'm at 5 to 10 kilojoules per mole. So if you have a very sensitive binding or something, it's not certain to be correct. So what I showed in the previous slide, how do we know that coarse graining is fine here? Well, we decided to test it. So this is this is again one of my ligand-gated ion channels, and it's a system binding a small molecule called PNU, a very strong potentiator. So we took just a handful of copies of these molecules, 10 of them in a gigantic system, and then we tried to sample where do these molecules spend time, which is not an ideal simulation, but we want to see, are you able to find the binding site if we don't know anything about the binding site? And now I'm just showing the protein here, and occasionally you see these red things fly by, and that's when these molecules are flying by. This is boring. Until boom. Do you see now that the molecule here has spontaneously found one of the binding sites? And you will actually keep seeing this through simulations, so that we have not used any of the information about the binding site, and yet in the simulation the molecules is able to identify a correct binding site. Again, it's not a guarantee. But when the simulation matches something you would predict from experiments, in general, the overall quality of the simulation is likely fine. And then I can probably also use the simulation to draw conclusions about the things I do not see in the experiment, because it is apparently able to reproduce things. And the latest state of the art we can do here is that Jan has simulated gigantic cells here. We've actually taken, this is this J. Craig Venter initiative. This is the smallest cell you can imagine. They basically took Mycobacterium and removed one gene at a time until they get down to under 500 genes. So this is a system within the ballpark of 200 nanometer radius and 8 billion atoms. And then we cheat a little bit, so we use this transmogrifier and shrink this. So that with coarse graining, this is in the ballpark of 80 million particles. And we can actually do a complete MD simulation of that. You're quiet. That's because you're in shock and awe about how amazing the biology is here, right? I'm a co-author of this. I can criticize ourselves a bit. To me, this is interesting as an academic exercise to see, can we scale things to very large systems? Can we get the codes to run on supercomputers? The problem is that we're not learning anything at all about the biology here. So this is exactly the problem I told you, right? It's really easy to push this button. The problem is that over 100 nanoseconds, there's absolutely nothing that happens in an entire cell. The end result after 100 nanoseconds is pretty much exactly what the starting result was. So we're going to try to push this up, and we might be able to reach a few microseconds or so, but long term, you're not going to be able to use MD simulations to study entire cells. We might be able, our idea is to use this to parameterize simpler models where we can propagate this, not even with particles, but with lattice models, and basically using the MD simulations to parameterize this. And that's the last memento mori I want to leave you with here, that just because you can simulate something doesn't mean that it's a good idea. And learn to resist this call to run a very large simulations and using 1,000 nodes or something. So that means that we're pretty much exactly half time here, and I'm going to move over a little bit to how we do things computationally. But if I reflect a little bit, reflection is good, and I would strongly encourage you to reflect on your own work looking back. There is a saying, to be a good scientist, actually not just scientist, to be successful in life, you need good judgment. You get good judgments from experience, and you get experience from bad judgment. Some of that there isn't really an alternative to, but one of the reasons with these summer schools is to be able to share some of our experiments so that you don't have to experience all those mistakes yourself. The single most important thing I should have learned 10 years earlier is to think before you simulate. The computer is an amazing tool, but it is not a brain prosthesis. If you don't know how you're going to analyze your systems after the simulation is done, then you're not ready to simulate. You need to have a plan. Why are you simulating it? And again, because you can is not a good answer. You need to have a rough idea of the timescales you're facing. Because again, there are processes in our cells that take an hour. And trying to simulate that with systems that the time steps is 1 femtosecond, forget about it. You're going to waste the next 12 months. And to be successful, you need to have, no matter how good your simulation is, maybe you're far better than I am. Maybe you have the perfect simulation. The only question is, how do you know? You might be convinced your simulation is great, but how are you going to convince others that your simulation or model, and this is equally true in bioinformatics, mind you. You need a way to convince other people that your model is accurate. And to me, the one obvious thing is that you need, in particular, if you're going to focus on computers, you need to be really good at understanding experiments. Because those experiments, you don't necessarily need to do them yourself, but you will need to constantly compare to them, interpret them, be able to argue that this simulation result actually supports that experiment. Or worse, sometimes you need to recognize that, oh, that's bad, but there are 3 important experiments here and they actually contradict my results. That happens. It's not fun. But the only thing that's even sadder is that if you ignore that. If there are plenty of experiments contradicting your results, there is something wrong with your model. And the earlier you accept that, the earlier you can fix it. Throwing more compute cycles on it is not going to fix it. And again, anything you know, if you know something about emotion, or you know that it's a part of a protein moving, you can get away from this brute force sampling. Maybe you can add 2 of these collective variables. Maybe you don't even need to do simulations. Maybe you just need, if you need 100 different candidate models, you might be able to use generative AI. It doesn't mean that you should not do simulations, but my team, we frequently combine one part of cryo-EM, and then we're generating 1,000 models with AlphaFold, and then we're using simulations to fit all those 1,000 models to various cryo-EM structures, and then check which one fits better. So just because you started by learning one tool, don't limit yourself to that tool. You don't need to master every single tool, but it's a really good ability to be able to follow what happens in the literature and use new tools as they come out. And the single point is the most important one. When you wear the life scientist hat, if I sit in my department of biochemistry and biophysics with one of my affiliations, there's only one thing I get credit for there, and that's making advances on biological problems. It does not matter how many flops I used. It does not matter what AI method I used. It does not matter how far I scaled. It does not matter if I use 90% of the available power of the computer. They don't care. If it does not solve an important biological problem, it's not an interesting result to them. Now, that sounds harsh, but if you're aware of that, you can of course design your studies so that you solve an important biological problem. And if you're in a life science department, this is key.